video we'll be looking at enzyme inhibition. An inhibitor is a chemical that could bind to the enzyme in a certain way to reduce the rate of chemical reaction. And there are actually, generally speaking, two more common types of inhibition here, and that's what we're going to be thinking about today. So first of all here, this is the structure of the enzyme, and we've got the substrate there. Now, if you look at the enzyme, there are actually two particular parts on the enzyme where the chemicals can bind to. The first one is this particular one with the V shape, which is the active site. The active site is the structure on the enzyme where the substrate would bind to have a chemical reaction happening. So that's the one of the sites on the enzyme. Now the other one, on the one below it, is called the allosteric site. So the allosteric site is a particular part on the enzyme which is in the active site, so that's not where the substrate binds to, but it's a place where other chemicals could potentially bind to. So these are two different places, and the two different types of inhibitors would bind to these two different sites on the enzyme to inhibit the enzyme action. So the first one that we'll look at is called the competitive inhibitor. So in the case of a competitive inhibitor, the chemical itself actually would have a very similar structure or very similar shape as the substrate itself. So for example, in this particular case, it might look something like this. So what we're saying in this case is that the active site shape is unchanged, right? So it doesn't change at all, but the thing is the active site is actually can also be bound by the inhibitor rather than the substrate. So they're both competing for the same active site, hence why it's called competitive inhibitor. Now the other one is called the non-competitive inhibitor, and the non-competitive inhibitor would look something like this, where it has a complementary shape to the allosteric site, but not the active site. But the thing is, the way that it works is that when the inhibitor actually binds to the allosteric site, the binding will cause a conformational change. It will cause a structural change to the active site of the enzyme. So for example, it will poke out something like this. Now in this particular case, the active site shape is changed and it's no longer complementary and specific to the substrate. So therefore the substrate cannot bind in this case. But it is not competing for the same site, hence it's called the non-competitive inhibitor. So what we'll do now is to have a look at the graphs, how the graphs would change if we're looking at the substrate concentration and the rate of enzyme action with or without these different types of inhibitors. So here we've got the axis, the x-axis has substrate concentration, and on the y-axis we have the rate of enzyme action. Now in the case where we don't have an inhibitor, the graph would look something like this. Right? As you increase the substrate concentration, the rate would also increase, and then eventually it would plateau because all of the active sites are completely occupied. So even if you add more substrates to it, the enzyme's already working at Vmax, the maximum capacity, the maximum rate of reaction. So this is without the competitive inhibitor. Now in the case where we do add one, then because obviously the inhibitor would slow down a chemical reaction, so the general rate would be lower than when we don't have one. The thing is, if you keep adding enough or keep adding more substrates to it, then eventually the chemical reaction would also still reach the same Vmax. So let's have a think about why that is the case. Now first of all, in the first bit of the graph, it's important to remember that the competitive inhibitor have a similar structure to the substrate. So for example, the V shape sort of structure there. Therefore, they can compete for the same active site, meaning that when the inhibitor has occupied the active site, there will be less active sites available for the substrate to bind. Therefore, there will be less enzyme-substrate complexes formed. Therefore, the rate would decrease. It's really important that you mention how more or less of the enzyme-substrate complexes are formed, as that is a really important marking point in the exam. Now, however, as we progress with adding more substrate to the actual mixture or chemical reaction there, because there's more substrates in the within that solution, that means that there is a higher chance for the substrate to actually successfully bind to the active site, because it's all about random collisions. When there's more of it, there's a higher chance of them colliding properly for the enzyme complexes to be formed. So therefore, the rate would also be restored back to Vmax. And in this case, we say the substrate can actually outcompete the competitive inhibitor for successfully binding to the active site, restoring the Vmax. And this is how competitive inhibition actually works. Now let's have a look at the non-competitive inhibition. So remember, the non-competitive inhibitor is where it can bind to the allosteric site instead. So again, if we start with having a graph where it doesn't have any inhibitor, so that's what the graph would look like, so exactly the same as the initial one before. But this time with the binding of the non-competitive inhibitor to the allosteric site, the shape of the graph looks something like this. Now in the case where the non-competitive inhibitor binds, because it binds to the allosteric site and not the active site, it actually is able to change the 3D structure of the active site. So in some sense you can say it changes the tertiary structure of the active site. So therefore we've got less of the active sites around that are complementary to the substrate, meaning less of the enzyme substrate complexes can be formed, hence the rate would drop down. But however, you can see in this case, even if you increase the substrate concentration, the Vmax is already lowered and it doesn't get restored. And that's because they're not competing for the same active site. So the fact that they are both binding to two completely different places on the enzyme, so once that is bound in, they have permanently changed the structure of the active site. So it doesn't matter if you add in more substrates to it, the active site is still changed by the non-competitive inhibitor. So nothing will actually change. So in some sense, the only way to really restore the Vmax is by somehow removing the non-competitive inhibitor. So let's go through everything as a quick recap. So this is the enzyme which has the active site that binds to the substrate and is complementary to the substrate. And it's also got an allosteric site, which is a site on the enzyme where substrates do not bind to. So we say how we have the competitive inhibitor, which has a similar shape as the substrate and can bind to the active site. So the active site shape isn't changed, but it's just being occupied. And in this case, the substrate can outcompete the competitive inhibitor by increasing its concentration. On the other hand, we have the non-competitive inhibitor, which can bind to the allosteric site. So in this case, the binding of it will cause the shape of the active site to change, and therefore the substrate cannot actually fit into the active site because it is no longer complementary to it. And in this case, even if you increase the substrate concentration, it will not restore the Vmax, as the active site is just completely altered, and they're going for different sites. And this is all about enzyme inhibition.